All right, so in this section, we're going to talk about the roles of and the different types of lipoproteins. It's important to have a good understanding of these as we move forward. And so we've talked about the chylomicron in terms of it being formed from the food that we've eaten. It comes from the small intestine and moves its way into the blood, as we just talked about. And it's made up of 99% lipid, and this is a picture of the chylomicron. Most of that lipid is triglyceride with a little cholesterol and the phospholipid cell membrane, a little bit of protein. Well, we also have three other types of lipoproteins. The first is the VLDL, or the very low density lipoprotein. This is formed in between meals, and it carries mainly triglycerides from the liver to the tissues of the body, and that usually occurs in between meals. It also has what's called an ApoB100. If you remember, we previously talked about what apolipoproteins are. Again, if my fist was a apolipoprotein, my fist was a lipoprotein, wrapped around it would be the apolipoprotein. So again, the lipoprotein would be my fist, and the protein wrapped around it would be the apolipoprotein. You need to have the apolipoprotein in order for the rest of the lipoprotein to be functioning. Well, you have different types of apolipoproteins. For the chylomicron, it is an ApoB48, and then for the VLDL, it's an ApoB100. Well, likewise, the LDL also has an ApoB100, but it's made up of a little bit different consistency. Mainly, if you notice that it has a low amount of triglyceride, but has a high amount of cholesterol. So the main job of LDL is to transport cholesterol to the cells of the body. So that is its role. Now, LDL gets a bad reputation for being the bad cholesterol, but it does have an important role of transporting cholesterol to the cells. The problem is when we get too many LDLs circulating, that it can cause disease. The final lipoprotein we're going to talk about is the HDL. Its main job is to transport excess cholesterol from the cells or from the cells or from the blood back to the liver. It basically cleans out and gets the excess cholesterol out of the blood and gets it back to the liver. It has an ApoA1 as its apolipoprotein. So these are the different makeups of the different types of lipoproteins. Now we're going to watch a video. It's pretty dense, but it talks about the roles of apolipoproteins and the importance of each. They're going to talk about other apolipoproteins, but the ones that we just are listed here are the ones that you need to be aware of. But it talks about the important role that apolipoproteins plays in the function of lipoproteins. So this 9-minute video is going to talk about lipoprotein metabolism, the different roles of lipoproteins. Again, you don't need to memorize all the different apolipoproteins, just the one that we just talked about. And the reason we're talking about apolipoproteins, again, you have to have them as part of the rest of lipoprotein to function. And also their role is they help to attach to the various receptors on the tissues of the body and help for them to function properly. So watch this video, and then we'll talk a little bit about it afterwards.
Reducing atherogenic burden is one of the greatest challenges for today's physician facing the epidemic of atherosclerosis. Fortunately, research is yielding enormous insights to meet this challenge. Recent decades of study have proven that lowering LDL cholesterol reduces cardiovascular disease. Yet still, many cardiovascular events occur without significant elevation of LDL cholesterol. Newer data are revealing significant atherogenic potential in lipoprotein particles other than LDL. In some circumstances, the composition of any of the lipoprotein particles can be altered, allowing participation in the atherogenic process. Furthermore, it has been shown that hypertriglyceridemia is a marker for the existence of such atherogenic particles. Hypertriglyceridemic patients at risk exhibit elevated triglycerides and low HDL cholesterol. This profile is often associated with the metabolic syndrome and diabetes, resulting from overnutrition, obesity, and physical inactivity, and leading to premature atherosclerosis. The epidemic of metabolic syndrome is expanding the high-risk patient population and has led to changes in therapeutic guidelines. In even newer research, the focus for reducing the atherogenic burden is moving away from the plasma and into the artery wall, specifically to the macrophage. Of great significance is the discovery of the receptors and transporters responsible for getting cholesterol into and out of the macrophage. These advances in scientific understanding are allowing yet another avenue for achieving our therapeutic goal: reduction of cholesterol in the macrophage of the artery wall. The treatment of dyslipidemia is continuing to evolve from a science previously grounded in the measurement of the lipid cargo, that is, total cholesterol and triglycerides in the plasma, to a science focused on the more sophisticated determination of the functionality of lipid transport pathways. Research is revealing complex interrelationships between lipoprotein particles, receptors, and enzymes necessary for functional lipid distribution. To understand these processes, let's first take a look at the dynamic structure and composition of the various lipoprotein particles, which serve as the transporters of lipids from sites of absorption or synthesis to sites of utilization, or in the case of atherogenesis, sites of accumulation. Lipoproteins function to package insoluble lipids for transportation in the blood. Two essential but distinct lipid types are carried primarily in the lipoprotein core. One type, the triglycerides, is an essential energy source, and the other type, cholesterol, is essential for cell membrane, bile acid, and steroid hormone production. Cholesterol in the lipoprotein's core is esterified. Unesterified, or free cholesterol, is enveloped in an outer phospholipid monolayer. Woven around and through the exterior layer are proteins called apolipoproteins. New data are proving these apolipoproteins to be more influential in lipoprotein metabolism than was ever previously recognized. The function of apolipoproteins falls into four categories. They are involved in assembly of lipoproteins, provide structural integrity, serve as co-activators of enzymes, and act as receptor ligands for cellular uptake. The unique collection of apolipoproteins on each lipoprotein surface indicates its origin, function, destination, and ultimate fate. For example, in the intestine, apolipoprotein B48 is involved in assembly and packaging of ingested triglycerides and cholesterol into chylomicrons. ApoB48 also provides this large, very triglyceride-rich lipoprotein with structural support. ApoE serves as the ligand for hepatic uptake of the chylomicron. Additional apolipoproteins, such as C2 and C3, may be acquired to regulate triglyceride metabolism. Particles remaining after triglyceride release, referred to as chylomicron remnants, retain ApoB48 and ApoE. ApoE is essential for hepatic uptake. In the liver, VLDL is assembled with apolipoprotein B100. ApoB100 provides structural integrity to VLDL and serves as a ligand for cellular reuptake. ApoE is another ligand for reuptake. As with chylomicrons, the additional apolipoproteins C2 and C3 may be acquired to regulate triglyceride metabolism. The smaller particle resulting from VLDL metabolism, IDL, retains ApoB100 and ApoE for receptor reuptake. The even smaller LDL, a remnant of IDL, retains only ApoB100. It is important to note that the lineages of particles just discussed all have ApoB in common, whereas HDL, The particle involved in reverse cholesterol transport features ApoA1 as its primary structural apolipoprotein. ApoA1 also plays a role in enzymatic reactions. In addition, HDL participates in carrying ApoA2, ApoE, ApoC2, and C3. These are referred to as exchangeable apolipoproteins because they readily transfer back and forth between HDL and the ApoB particles. Revelations about the unique combination and function of the apolipoproteins on each particle are continuing to advance our knowledge of lipoprotein dynamics and the distribution of their cholesterol and triglyceride content. Accordingly, our evaluation of fasting plasma lipids continues to evolve. Over the years, guidelines have shifted from the measurement of total cholesterol and triglyceride content of lipoproteins in the plasma to a more refined focus on LDL, the bad cholesterol, which deposits excess cholesterol in the arterial wall, and HDL, the good cholesterol, which retrieves cholesterol from the arterial wall and returns it to the liver. While LDL is clearly recognized as the major atherogenic particle, new evidence indicates that increased levels of other triglyceride-rich lipoproteins, including IDL, VLDL, and chylomicron remnants, also contribute to cholesterol accumulation in the arterial wall. Together with LDL, these atherogenic lipoproteins are defined as non-HDL. In patients with a predominance of non-HDL, triglycerides are elevated, and in these patients, current guidelines recommend the calculation of non-HDL cholesterol, which reflects the bad plasma cholesterol from all atherogenic lipoproteins. Non-HDL cholesterol is equal to total cholesterol minus HDL cholesterol. Furthermore, it's important to remember that non-HDL and HDL can also be distinguished according to apolipoprotein content. For example, ApoB100 and ApoB48 are unique to non-HDL, whereas ApoA1 is unique to HDL particles. As the science progresses, the routine measurement of plasma apolipoprotein concentrations may guide clinicians into even more sophisticated decisions regarding dyslipidemia diagnosis and management.
Okay, so that was a video that did an overview of the lipoprotein metabolism, apolipoproteins. Again, you don't need to memorize all the different ones that we talked about, or they talked about in the video, but just the ones that we talked about at before the video started. But it was good to have a good understanding of at least how they all look, what their appearance are, and then now we're going to talk about how the roles of each of these in the lipoprotein metabolism as we move forward. So again, this is just their overview of what we just talked about, and also the video talked about. So just again, moving forward, here again is an overview of the chylomicrons and the VLDL. The key being that they both transport triglycerides. The difference is again that chylomicrons are transporting triglycerides from the meals we've eaten, and they're formed from the meals we've eaten, whereas VLDL are between meals. And we're going to show the role of VLDLs in just a bit. And this is just the difference between the classes. We have the chylomicrons, VLDLs, and the LDLs, which are pro-inflammatory, meaning causing disease, and the HDL is anti-inflammatory because of its role in taking up excess cholesterol from the tissues and getting it back to the liver. Also taking up excess cholesterol from the blood and getting it back to the liver as well. Here's the how you measure the different lipoproteins. You may have gone to the doctor, had a blood draw performed, Here is the blood draw. They take it, they put it in a tube. It gets spun down, and then upon spinning, you have the HDL, LDL, and then you have the IDL, which is the intermediate density lipoprotein, which we won't talk too much about. And then you have the VLDL. Well, these can actually be parsed out into subfractions or fractions. And what's important about these, as we're going to learn later, is that not all LDLs are created equally. So you have actually large, dense LDLs and small, dense LDLs. And we're going to talk about why there's a difference and why the role of each, why one may be unhealthier than the other. One other thing you need to consider is the amount of cholesterol. Which of the lipoproteins carries the most cholesterol? Well, this is the amount up here. The higher the cholesterol indicates here that the highest amount of cholesterol is found in LDL, followed by HDL. So you'll find out on the exam that which of the lipoproteins contains the most cholesterol. Well, it's LDL. All right, so this concludes the portion about the lipoproteins. Make sure you have a good understanding of the various ones. And as we move forward, we're going to talk about now the roles that each of these have and how they're made in the body.